Hi everyone, today we are going to share with you about a biomedical engineering course named Biomedical Materials and Mechanics Experiments. These are our team members. Our content mainly consists of course introduction, hydrogels experiment, finite element analysis experiment, some basic experiment techniques, and lastly our earnings from this course. Let's start with the course introduction. First of all, this course is to let us implement useful skills or concepts in various fields of biomedical engineering. It consists of different kinds of experiment. For the mechanics of materials part, we study the operation of MTS. Secondly, for the use of finite element analysis software, we use software such as SolidWorks to draw bones, bone nails, and bone plates, and then use ANSYS to simulate and analyze the force of each other under such configuration. For the observation of biocompatibility, different kinds of cells are used for testing, cell culture skills, counting, dyeing, and finally for the suture after implantation in animals, we learn various types of sutures. These are some skills that may be required when doing such experiments. Therefore, in this course, it allows us to briefly experience and learn these experimental techniques. At the end of the semester, we will also have a group project report, and the theme is based on the techniques we learned in this semester. It trains our ability to think independently and deal with problems just like during research and topics.
So this part is about hydrogel preparation. Preparation of photo-cross-linking hydrogel and temperature-sensitive hydrogel are the main knowledge we learn in this part. So we use ketone, acetic acid solution AA, napalm, APS, and TMIC for temperature-sensitive hydrogel and control the temperature to determine the mechanical properties of hydrogel. Besides ketone, AA, glycidic methacrylate GMA, and LAP, which is the photoinitiator, are materials of photo-cross-linking hydrogel. We measure different mechanical properties while controlling different intensities of UV light and time of exposure. In our final project, we solve photo-cross-linking hydrogel and determine its mechanical properties through MTS test and swirling ratio. First is MTS test. After we prepare the hydrogel, we directly salt it into different concentration of MgCl2 solution for 6 hours, then compress its gel and calculate strength and Young modulus of each. Next is test of swirling ratio through weighing each hydrogel before and after soaking. As a step of MTS test, we soak hydrogel directly after preparation. After freezing and freeze drying, we soak hydrogel in SPF solution with different duration and measure their weights for 3 times. So after calculation and comparison, we found that hydrogel will have higher compression strength and swirling ratio after soaking in MgCl2 solution. As summary, we not only learn the theory of cross-linking of hydrogel, but also know how to do it by hand. Materials preparing and mixing, solidifying, dialysis, freezing, and doing MTS tests can all be learned in this part.
I'm here to tell you about the finite element course. In this section, we have learned to use the finite element method to study the stresses between a structure and predict the deformation or failure of this structure under unknown stresses. For example, in our final project, we have used finite element analysis to examine the deformation of the lumbar spine on a dynamic and a static stabilization device. With SolidWorks, we first generate the complex models, which include the lumbar spine fixed with dynamic and static stabilization devices in different positions, as shown in the figures. After that, we use ANSYS to solve and preview the lumbar spine model's deformation. Finally, we can wrap up our research with the various analysis results. For instance, we discovered that the lumbar spine model has the minimum deformation when a static stabilization device is fixed on L3 and L5 vertebrae, or a dynamic device is fixed on L3 and L4 vertebrae. In summary, we have learned the entire finite element analysis procedure, including how to build a model that we want to analyze using SolidWorks, and how to solve and predict the stress as well as deformations of the model with ANSYS. This concludes the finite element section.
When it comes to the biomaterials and biomechanics experiments, there are some fundamental experimental skills I want to share. At first, I would like to introduce the simple interrupted suture. Because of its simple operation and not affect ejection sutures, it was used to deal with infected wounds. This kind of suture is usually applied in animal studies. For example, artificial cartilage implants in rabbits for compatibility testing. Next, we move to the vertical matrix suture. It is used in folded anatomical parts, especially for the loose skin of the pregnant women. Although this is good for the prevention of issue bleeding and better recovery, it is easy to cause scars. So that's the reason why the postpartum women have obvious scars on their belly. Next, horizontal matrix suture is often used for the anastomosis of the vascular wall, or the closure of peritoneum and pleura. It features with little influence on blood circulation of wound margin and prevention of issue bleeding and better repairments. However, it is time-consuming and make issue compressed if stretched too tight. Then, simple running suture is different from the previous sutures. It is easier and causes little scars but tend to wrinkle. The most appears when the low width needs to be sutured. Compared to last suture, simple rounding locking suture is equipped with high tensile strength and good hemostatic effects. Therefore, it is suitable for the wounds with good blood circulation and additional hemostasis. After the suture skills, cell culture is also critical in biomaterials. Cell culture is to simulate the body physiological environments in vitro for any conditions of experiments. It is convenient and economically in large quantities for experimental samples. Next, cytotoxicity test is extremely common in biomaterials as well, especially when it comes to testing whether the experimental metallic ions will be harmful to our health or not after the metal implant is in your body for a while. Frozen section is a useful technique to increase the stiffness of the issue to provide the support for sectioning. Its thickness can be close to 10 micrometers, which means it is easy to stain for further analysis. Finally, HE stain is classical staining method in issue engineering. Hematine, as the oxidation product of hematoxylin, has the characteristics of an indicator. When it fails to acid conditions, it turns into reddish. Otherwise, in the opposite conditions, it turned blue. As the figure shows, according to this phenomenon, the nucleic acid will be red and other places without acid will be blue.
The last is our earnings from this course. This is a highly recommended course for everyone because we can learn many basic experimental skills, and this course is allowing us to study in a relaxed and fun atmosphere. Hydrogel is the most challenging skill because it is difficult to determine whether there is an error in this final project as it is a chemical reaction. But dissolving and solidifying the hydrogel solid is a very interesting preparation process. The most memorable part of this course is the suture skills. It has 5 teaching methods in this course. Besides, we also learn the final element analysis software. We use SolidWorks to build the model and then use the ANSYS to analyze our model. Thanks for your listening.